So it's my great pleasure to introduce Jasmine Foo. Jasmine works in things about tumor evolution using stochastic processes of lots of different sorts. And her work comes from a background in, her PhD was in applied mathematics at Brown, and her undergrad was at Brown as well in mathematics and physics. She was a postdoc at Memorial Sloan Kettering and Dana-Farber, I think. And she's been at the University of Minnesota since 2011, where she's now the Northrop Professor of Mathematics and the co-director of the Therapy Modeling and Design Center at the University of Minnesota Twin Cities. So I'm really interested in the sort of work Jasmine does, which is a stochastic processes jock, which used to be what I was as well. So I'm looking forward to hearing about how computational methods work. Jasmine, welcome to IICD and many thanks for coming.
Thank you so much for the invitation, and I'm really happy to be able to be here and talk with many students and faculty and tell you a little bit about what we've been working on in the past couple of years in my group. So this is a quick outline of the talk. So it'll be a two-part talk, and generally the topic is tumor heterogeneity. In the first part, I will be talking about profiling, some methods for profiling drug response heterogeneity using bulk tumor drug screening data. And the second part, we'll be taking a little bit of an evolutionary modeling perspective for interpreting genomic site frequency spectra. So the first thing I want to talk about is high throughput drug screening. What do we mean by this? This is an important tool in personalized oncology today. Generally, what happens is that we take patient tumor samples and culture them in various kinds of experimental platforms. So this could be here shown 2D cell culture, but also there are lots of interesting drug screening studies going on in 3D experimental platforms that more accurately capture the tumor environment, such as organoids or microtissues. And the idea, of course, is to treat these samples with many different kinds of therapies and use that information to help inform personalized treatment decisions. So the kind of data we typically get out of a drug screen might be something like this. So you have a cell viability versus drug concentration. So this is the drug response curve. And viability has several different definitions in the literature. This is one of them. So at a particular drug dose D, at time T, just sort of recognizing that all these experiments are done in the same time period, 72 hours or something like that. The viability would be the cell count at that endpoint time T under dose D, normalized by the cell count at time T with no drug, for example. And I want to make the comment that just like there are various experimental platforms for culturing the samples, there are also different kinds of experimental quantitation assays to get these cell counts. So things like tetrazoline reduction assays, MTT, MTS, ATP assays, and things like real-time glow or AnkiSite. And the point I want to make about this is that some of these assays are endpoint assays where different data from different time points actually reflects separate experiments because the cells need to be lysed at the time of counting. Whereas some of the newer assays are sort of life cell imaging, so you can actually track the same population over time. And the reason I bring that up is because that has sort of interesting features or consequences that we want to think about when we build sort of noise models into our analyses. Okay, so the major question that we're or the major challenge that we're interested in is the fact that Of course, you all know here that tumors are actually very heterogeneous, and in fact, there may be many different cell types, each of which responds to drug in a different way. So when you get a readout of cell viability or drug response, you're actually looking at some sort of mixture response to drug, which may not represent the sensitivity profile of any individual population in the tumor. Okay, so this, of course, complicates things, and if there are heterogeneous, if there's heterogeneity in the tumor sample, then you may have, for example, small resistant populations that become very clinically relevant during treatment and drive tumor recurrence. So what we are interested in this project is, can we detect subpopulations from bulk drug response data coming out of these drug screens? Can we quantify how large or small those subpopulations are? And can we understand what is the drug sensitivity profile of each of those population subcomponents? And I'll just mention here that we're going to use a particular sort of summary metric of something very related to the cell viability curve called the GR50. I'm not going to get into the details in this talk, but I'm happy to answer questions about it. But if you see me saying GR50, that's sort of a summary statistic of this drug response profile, if you will. Okay, so how do we do this? I'm going to start out with, for the purposes of simplicity, I'm going to be talking about a very simple situation where we're going to think about a mathematical model for population growth within one of these experimental cell culture platforms in which the drug response assay is being done. And in particular, we're going to consider the simple case where we have cell types that don't interact with each other, and we're thinking about some 2D cell culture. So this is the simplest setting. So you can think of, to say, a simple exponential growth in the setting where we just have one population. So we have x of td is the population size at time t under drug concentration d. Starts out some initial size, grows exponentially, and we have a growth rate that depends on the drug concentration d, and alpha is the intrinsic cell growth rate in the absence of drug. And h of d has this sort of classic hill type form, which is why I still have this figure up here, where we have a couple of parameters governing the shape of this function. B represents sort of the maximal effect, or where this thing's going to level off. E tells us where this decline is, the log concentration at which 50% effect is achieved. And then N tells us kind of the steepness of this growth rate decline. Then building from this, we can build up a model of polyclonal population dynamics. Again, remember, these are non-interacting, so we simply have the sum of these populations. In this case, S is the total number of populations. Z is our total population size. So the readout we're getting from the drug response, the drug screen assay, is some function of Z, and pi sub I is the initial frequency of the ith subpopulation at the beginning of the drug screen. Because of course the frequencies of these populations change during the course of the drug screening time period. And I should say that this work was really driven by Alvaro Cohn-Luke and Evan Mikolhus at the University of Oslo. Okay, so theta S is the set of parameters that we are interested in estimating, the initial frequencies of each population. the intrinsic cell growth rate, and then these parameters of the drug response profile for each of the S subpopulations. And of course, we don't know what S is either. So that's also something that we need to estimate. Okay, so using that basic model, we can go and think about, what's the data that we get out of these drug screens. So the observed data is some sort of total cell count under a series of drug concentrations, EJ, time points, TK, and we have some replicates, some number of replicates for each dose time point pair. Was there a question?
So do you expect this total cell count to be the data or fraction because it's you're sampling out of the entire tissue?
So that's a good point. When I say total cell count here, I am talking about the total cell count within the experimental drug screen assay, which is taken from a sample of the tumor. Okay, so this is a model of the experimental assay, not of the tumor. Any other questions? Okay, so we're gonna our model of the observed data is that we have our underlying population dynamic model, this Z from the previous slide, plus some Gaussian noise term. Just a little note on the Gaussian noise. From the data that we have, we can see that there is variation in the standard deviation. So motivated by that, we introduce a sort of bi-level standard deviation structure. We have high and low variance depending on the dose and time point. So we have at lower drug doses and higher time points, a higher variance, and sort of these thresholds, as well as sigma H and sigma L, become our 4 parameters of our standard deviation. And then we can write down a log likelihood for our model. And you can see the 2 pairs of terms coming from this standard deviation structure.
So just going back to the standard deviation structure, when you're saying that you have for lower densities, you have different sigmas, is that because you're not accounting for quorum sensing, or because you're not assuming local interactions?
That's a good question. I'm going to actually, in a couple slides, introduce a second model which addresses this particular question and I think has a better, more sophisticated model of the standard deviation that where it kind of arises more naturally and more flexibly. But to answer your question briefly now, the motivation for this structure was that we could just simply see that in the data that at lower doses and later on in the experiments, the data points had higher variance.
So is that, so you can, one may also argue against just like density dependent effects instead of actual radiation, right? You could have a biology lift effect because of cell densities vary in cancer cultures, like among the
yeah. I mean, so I guess I'll say that we have incorporated, I was mentioning that our model, the model I showed on the previous slide, was the sort of simplest setting where we have non-interacting populations. We have also formulated versions of this with interacting populations and density dependent effects and plasticity and things like that. For the particular data sets that I will show you here, I believe the variation in the standard deviation is due to demographic stochasticity, and thus that signals the need for an underlying stochastic model of the population dynamics rather than a deterministic. And so I'll sort of show how that captures the patterns in the standard deviation that reflect what we saw in the data.
Does replication mean separate samples from the same tumor? It's not exactly the same group of cells, right?
That's right. Well, yeah, so separate samples that are originally from the same tumor.
They can have different mixtures of cell types and genotypes.
Yes. Although, sorry, I'm hesitating here because most of the experimental data I'm going to show you is actually controlled admixture of admixtures of cell lines. But yeah, that is something that so I guess you're taking a sample from the tumor and then you're sort of parceling it out into different wells, for example. So you could end up with slightly different compositions. Okay, so the overall algorithm is to maximize this log likelihood to estimate this set of parameters for varying subpopulation levels, and then to estimate the number of subpopulations via some sort of model selection, so information criteria, for example. And then the algorithm, which we call phenopod, returns the estimated number of subpopulations, frequencies, and the growth characteristics. That's this GR50, so drug sensitivity of each subpopulation. Okay, so we first started trying this on simulated drug screening data. So we created in silico 3 cell lines and mixed them at various mixture fractions. And we added standard deviation of noise 5% of the initial cell population. What you're looking at, just to kind of acclimate you to the kind of graphs I'll be showing, is on the left column here, this is the sort of known truth about the mixture frequencies. So this is a mono population, this is 3 populations at these frequencies. On the right is what the algorithm spits out for mixture fractions. This is at the start of the drug screen. And then over here, these are the drug sensitivity metric GR50. The x-axis is drug concentration. So the sort of lighter colored bar is the true GR50, and then the darker band is what the algorithm is estimating. So you can see that in the sort of simulated data, where our data is generated from the model that I told you about, then it's doing pretty well at both estimating the mixture fractions. The number of subpopulations is correct, and the drug sensitivity metrics also are pretty accurate. Then we moved on to in vitro data. So this is experiments were done by Dagan Tadali. He's now at Cleveland Clinic, and he was in Shabtal Tassin's lab at the University of Oslo. And so this was, in these experiments, they mixed known mixtures of these cell lines with differential response to imatinib. So they were isogenic with and without the T315I mutation, which is sort of known cause of resistance to imatinib in BCR-ABL driven or BCR-ABL cell lines. So here we can see again that so we took the bulk drug response data from their experiments and then used it to estimate or to output a heterogeneity profile, which ended up matching well with both the mixture fractions as well as the drug sensitivities. Independently, we had another set of data from Shannon Momenthaler at Ellison, who did some admixtures of non-small cell lung cancer lines with differential response to erlotinib, another tyrosine kinase inhibitor. So these are cell lines with and without the T790M mutation. And so this was an independent lab, different experimental assay for quantitation. Also the method did pretty well at estimating the mixture fraction, the number of subpopulations, and the drug sensitivity. Okay, so next we wanted to think about the effect of experimental noise. So if we generate this data in silico and we turn up the measurement noise, how far can we go before this thing starts to break? So here on the left is 10% noise, on the right is 20% noise. I don't mean for you to parse everything about these graphs, but just sort of overall, we can see that, as we go to 20% noise, we're getting some inaccuracies in terms of the drug sensitivity. But overall, it looks okay. And remarkably, the mixture fraction also looks pretty good for most of these cases. What kind of noise is normal for common experimental techniques? We did a little bit of a literature search and also looked through the data that we got from our collaborators. And the general consensus in the literature was anywhere from 1 to 15% was sort of estimated noise. Okay, so then we also looked at some clinical samples in multiple myeloma. Of course, now we don't know the ground truth for the heterogeneity profile, but so multiple myeloma is a malignancy of white blood cells involving an abnormal proliferation of these cells in the bone marrow. It's sort of characterized by patients who patient clinical histories with sort of multiple recurrent episodes of remission and relapse on different therapies. And it's sort of known that there's sort of these complex clonal dynamics that change with different courses of therapy and sort of drive an unpredictable treatment response. So again, collaborators at University of Oslo did this work of taking these samples from patients, culturing them, and then putting them through a drug sensitivity assay in these 384 well plates. This is a 96-hour drug screen. And here are some of the results. And these are some multiple myeloma patients treated with 6 different drugs here. Not every patient was treated with the same set of drugs, which is why you see different numbers of rows here. So what you're looking at is the same sort of plot where each panel corresponds to a different therapy and each row or color corresponds to a different patient. So what do we take away from this? I think it's interesting that for some therapies, you have very homogeneous sort of responses between patients. So for example, for this therapy, we've got all the patients with sort of the same one clone being the same, and this one patient has sort of 2 clones. Whereas there are other drugs for which sort of the response is all over the place between patients. If we look at one therapy, this therapy here, venetoclax, we thought it was interesting that there were sort of patients with kind of similar clones that grew at similar rates or had similar responses to therapy, but maybe at different frequencies. And so we actually looked in those 2 samples for the presence of, for this particular drug, is sort of some known high-risk genomic abnormalities that are associated with resistance, and found that they were present at basically the same frequencies in these 2 patients that were predicted by the algorithm. Okay, so and what do we do with, so once we have this drug response heterogeneity profile, what can we do with it? Sort of as I'm not going to get too much into the details here, but one of the things that I think is really exciting about what you might be able to do with those kinds of profiles is to use them as initial conditions for mathematical models for predicting treatment response on an individualized basis. So to sort of explain these plots, multiple myeloma cells produce an abnormal M protein, which somewhat tracks the disease burden or indicates some information about the disease burden. And so you can write down a mathematical model of the dynamics of multiple myeloma, in particular M protein readout. And so we did that, and we set as initial conditions the heterogeneity profile for these 3 patients. And then we kind of put them all in the model on the same venetoclax treatment regimen, and then kind of ran the model forward in time. And so, as I said, I'm not really going to go into the details of this model, but the sort of takeaway is that just sort of underscoring the idea that you might have patients with the same, even the same, the presence of the same clones in their disease, but being at different frequencies can drive pretty different treatment dynamics or treatment response dynamics. Okay, and let's see, how am I doing? Maybe I'll skip this. And I'll talk a little bit about some of the very recent work that we've done on this. That kind of comes back to this question of the variant structure of the model. So one of the things that we saw in the data was not so this sort of bi-level variant structure was kind of a simple solution to reflect this the fact that different data points had different standard deviation levels or different dose time, concentration time pairs had different variation levels or variance levels. But really what we saw was that we had continuous range of variances in our data with sort of linear dependence on the population size. And in addition, we wanted to have a more sophisticated model that could kind of speak to the fact that some of our data comes from live cell imaging, where you're really tracking the same population over time, and other data is coming from endpoint assays. So what we did was to replace the deterministic growth model with a stochastic linear birth death process model. Has a more complex variant structure, which reflects, which captures the data better, and also has the ability to handle the sort of correlation structure at different time points that we want. So to kind of describe this model, so once again, we have a total population. We observe some function of this, which is made up of S subpopulations. N is our initial population size. For each of the subpopulations, we use a linear birth death process with birth rate beta I and death rate, which has that same dependence on drug, hill type dependence on drug. So the mean of this model is going to be the same as the deterministic version, but the variance has this structure. And here we can see the sort of linear dependence on the population size that we kind of saw in the data as well, which to me suggests that it's coming from the sort of demographic stochasticity for the most part. And here, the parameters that we're estimating, we have one more parameter than we did in the previous model. In particular, we still have our drug response profile parameters for each of the subpopulations. And instead of having alpha, now we have beta and nu, sort of representing the decomposition of birth and death. Okay, so the calculation of the likelihood for that model, the stochastic model, is more difficult because X of t, this total population, is non-Markovian, and the complexity. So we really have to sort of sum up over all these different, hidden states, and the complexity is going to increase with the number of subpopulations. So what we did instead was to use to prove a central limit theorem for this process, and derive a limit, which is a Gaussian process, And then we work with this limiting approximation to derive the likelihood for live cell, both the live cell and endpoint assay data sets. So for example, for the live cell imaging type of data set, we have our likelihood where we're looking at now. So here I've dropped the dependence on drug because it will be this sort of you do this process for each drug concentration. So we have our different time points, so observations at different time points, and this is what our likelihood looks like. So here, mu is our mean, which depends on our initial frequencies and our net growth rate of the ith population, which is going to be lambda sub I. And then if we look at this sigma here, that's coming from this Gaussian process limit. It's a correlation matrix of this Gaussian process limit normalized by our tumor size n, or scaled by the tumor size n. Okay, so and it turns out that this improved method is better at detecting small resistant subpopulations. So we simulated some data sets with an unbalanced initial proportion of sensitive to resistant subpopulations. So on the x-axis is the initial proportion of the sensitive subtype. So mostly sensitive, small amount of resistant subpopulation, and on the y-axis is the error. And so you can kind of see that the original method, Phenopop, doesn't do so well for very small resistant subpopulations, but the formulation with the stochastic population dynamic model does a better job. The two lines are endpoint versus live cell image or real-time data sets, which I won't get into here. And this work was mostly driven by Chen-Yu Wu, who's a grad student at Minnesota right now. Okay, I will now move to the second part of my talk, which is about inferring tumor parameters from genomic site frequency spectra. Probably I don't need to describe this slide to anyone in this audience, but just real quickly, as you all know, there's sort of many different theories about how tumors evolve from kind of the very simple linear evolution, where you use sequential sort of selective sweeps, to sort of neutral evolution, where you're getting a lot of coexisting clones and many mutations coexisting, and everywhere in between. And and there's an interest in how can we use genomic data to infer things about how about a tumor's evolutionary history. And so the big challenge in this field, or at least from my point of view, is that we have samples at a very limited number of time points, usually or many times only one time point. And a way forward, which is taken by many people in the community, is, can we use the richness of genomic the typo data that is observed at a single time point to infer evolutionary characteristics. For example, can we tell whether something's evolved under neutral evolution or selection? And can we infer quantitative parameters of the evolutionary process? And the site frequency spectrum is a popular summary statistic that is used to explore genomic data. And classically, it's been used for developing estimators of effective mutation rates in fixed size population models like Moran process and Wright-Fisher. Okay, a quick review of the site frequency spectrum for anyone who is not super familiar with it. Basically, you can think of this as a cell of origin for a tumor, and maybe it's picked up a mutation A, And then in the next generation picks up another mutation, B, and so on and so forth. Some of these cells die. The site frequency spectrum is a sequence of Sj of t, where Sj of t is the number of mutations present in J cells at time t. So in this example, let's say t is the present time at the bottom here. We're going to look across this set of cells. Well, there are 4 mutations that are found in exactly one cell, E, G, H, and I, and so on and so forth. Okay, so so the goal is to try and understand evolutionary history using the site frequency spectrum. I want to introduce a branching process model of evolution now. So cells in the model divide at rate R naught, they die at rate D naught, independence is assumed, and again, we sort of use this lambda parameter to delineate the net growth rate. Each time a cell divides, it's going to accumulate a random number of neutral mutations. The mean of this random variable is W. And just a little bit of notation that you'll see in the next couple of slides, the extinction probability of a clone derived from a single cell is P naught, which is D naught over R naught, known from branching process theory. Q naught is the complement survival probability. And just repeated the definition of the SFS, or site frequency spectrum there. Okay, so as I said, SFS has been studied, well studied in sort of classical estimators for effective mutation rate in fixed size populations. There's less work done in expanding populations. As far as I know, some of the earliest works were Direct 2013, and Kessler and Levine, and Bozich, Gerald and Novak in 2016. To kind of summarize some of these results, if we define M of F to be the number of mutations at a frequency F or larger, and N is the number of tumor cells at the sampling time, then we get this form for the expected value of M of F. We notice this 1 over F scaling here. And this is sort of the cumulative version of the SFS, analogous SFS result, which is the expected value of the number of mutations found in J cells, which has a 1 over J squared form. So these are sort of analogous results. One's a cumulative version of the other. Importantly, these results are based on an approximation that considers the only cells with infinite lines of descent, which we call the skeleton process. And I'll kind of sort of explain later why this is valid in only certain sections of the SFS. Applications of these results, Williams et al. Proposed a neutrality test based on this 1 over F law applied to genomic data, I believe in colorectal cancers, sort of a landmark paper. And there have also been other estimates of this effective mutation rate, also in this paper, as well as a few other papers. And this is by no means a complete characterization of the work that's been done on this. Okay, so what's our goal here? Well, the prior work focused on the large frequency end of the SFS. So this kind of pertains to the skeleton process approximation that I just mentioned, that I'll explain in detail a little bit more. So we'd like to obtain a more complete understanding of the SFS across the entire spectrum of frequencies. And we will then use that information to develop a new approach for estimating these parameters separately using the small frequency end of the SFS. So one thing I forgot to mention is that, and we'll see how this pops out. Actually, you can see it right here, W over Q naught. These are coupled in this approximation. So it's not really possible to decouple these parameters using this 1 over F law. Okay, so we have some results on the SFS. across the spectrum. First, I will sort of focus on the fixed time SFS, in which we consider the population at this particular time t sub n, which is basically the time at which the expected population size hits level n, conditioned on the event where this thing has survived until that time. Okay, so if we look not just at the skeleton process, but the entire population, then we find the following. It's kind of a messy formula, but it's the expected site frequency spectrum, and it's valid across the spectrum. And let's see. To sort of simplify things, let's look at the large N limiting regime. So that's the regime that's relevant in cancer. We end up with this thing that sort of starts to look a little bit like the 1/j² law from the previous slide. In particular, if we set p equal to 0, so that's the no death, so then the entire tree is the skeleton process, then we're going to recover the previous results. Oops. Sorry. Okay, we also looked at the fixed time SFS, which considers the population when the population size hits n. So this is stochastic hitting time. The formulas get a lot more complicated, so I'm not going to put them up here. And I should mention this work was driven by Einar Gunnarsson, who's a postdoc now at University of Iceland. And so the fixed time SFS results are in the published work, but I will just say that for large N, they coincide with each other. Okay, so what's interesting about this? I've alluded to this a little bit earlier. At large frequencies, the SFS is approximated well by the 1/j² law, this sort of skeleton-based approximation. Right? So if we send in this large N limit, the SFS expected value, if we send j to infinity, then we do get this same result. However, we think about the rest of the spectrum, not at the large frequency end, we fix j, and we send p naught to 1, so that's the regime of high death, which is relevant in cancer, and has been sort of the subject of investigation in some other works, which I unfortunately don't have the references for right here, using sort of serial tumor data. P naught turns out to be sort of between 0.9 and 1. Then what we find is that this expected value of the SFS looks like the SFS of the fixed size population classical Moran model. So what's going on? So here is the SFS in the y-axis, and on the x-axis is j, the number of cells. So this is the large frequency end, this is the small frequency end. So and then the dashed lines, the red line is the 1/j² law that coincides with the skeleton approximation, and the green line is this fixed size population 1/j law. So what we see is this sort of transition at the large frequency end. This thing matches up well with the 1/j² law. Then as we move towards the low frequency end, it's sort of the spectrum bends towards this 1/j law. Right. So and this is the two plots are for different values of n. Both of these plots happen at large p naught, which is the relevant regime we're interested in. Okay, so to think about this, when we are looking at the large frequency end of the SFS, in order for mutations to get up to that high frequency, they have to have started early on, and they have to be on the skeleton so that they survive. So a sort of skeleton approximation is valid up here. whereas as we move towards the low frequency end, we start having to consider cell families that will die out. And as we move to the really low frequency end, in the regime of high death, we're looking at a lot of families that die out, and these families, their population behaves like a critical birth-death process, which is why this sort of Moran SFS starts to kick in. Okay, so what can we, so it turns out we can sort of use this deviation at the small frequency end to propose a way to estimate these evolutionary parameters. Okay, so let's let M sub 1 denote the total number of mutations in a sample. And so we can derive under the model that we have what we expect for M1. So expected value of M1, again, conditioning on survival until this time, Tn, this time at which the expected tumor size hits size n. And then let's consider S1, which is the sort of lowest frequency end of the SFS. So sort of theoretically, the lowest thing you could, you probably can't detect it at this point, but so it's the low frequency end of the SFS. The expected value of this thing we can also derive under our model. And the ratio in the large n limit, well, the ratio is independent of W, and then the large n limit looks like this. Right? And so it's a sort of independence of W that provides an inkling of, well, maybe we can develop an estimator here. Because remember, W and P naught were existed appeared only as a ratio in the previous estimate. And so to sort of flesh this out, let's we can note that this function, this ratio, which now we'll call a function of P naught, phi n of P naught, is actually decreasing and invertible. It maps 0, 1, this fraction, to this interval here. So if we take the inverse of phi and we plug in the expected S1 divided by the expected M1, then we should get out P naught. Okay, but you may be thinking, well, this is expected value. You're only looking at one tumor sample. So it turns out we can get a large law of large numbers result that basically tells us that in the large n limit, this ratio for a particular tumor sample will converge to this thing we're interested in. And this enables a consistent estimator of P naught based on data from an individual tumor. So that's our estimator. So we get S1, we get M1, we take the ratio, we invert this thing, and that's our estimated P naught value. So we tried this on simulated data. What you're looking at is the histogram of the estimate P naught hat for n equals, say that's a typo, 200 and 1000 for various values of P naught, 0.5, 0.7, 0.9. And so in all these cases, you can kind of see that this thing is narrowing around the true value of P naught. So the caveat here, of course, is that, you know, we are not yet at the point where we can actually obtain and we cannot observe reliably S1. It's a very low due to the detection limitations of the techniques. But I think you know, as the technology improves, we can also do some work on our end to push this forward to numbers that are higher than 1. Certainly signal will still exist there. And the question is, how much will it be diluted as you look a little bit farther out in the spectrum? Okay, so just to summarize, the first part, we developed this algorithm that profiled the phenotypic heterogeneity from bulk tumor drug screening data and applied it to a multiple myeloma data set. Importantly, it's based on flexible population dynamic models of heterogeneous subpopulations. We're currently extending this to different experimental culture platforms, such as organoids, and have been looking at interacting populations with plasticity, so where cells are interchanging states in time. We extended this to a stochastic model with a sort of better variant structure. And we hope that the resulting heterogeneity profiles can provide insights into the development of treatment strategies. And then for part two, we characterize this exact form of the SFS for fixed time and fixed size. Sorry, that sentence doesn't make a lot of sense. SFS for neutrally evolving tumors. We can kind of see this transition from a 1/J² law to 1/J law at small frequencies, as you go from large to small frequencies, and we can exploit that sort of bending of this SFS to develop sort of a new perspective on estimating the mutation rate and extinction probability. Most importantly, I need to acknowledge all the people who did this work, Einar Gunnarsson, and Einar Gunnarsson was involved, drove the SFS investigations along with Xuan Zhang. And then for the mixtures work, for the phenopop work, Alvaro and Evan and Chen Yu have all been really driving this work, and experiments for the deconvolution work were done primarily by Maria Serena de Liberto for the multiple myeloma samples, and Dagim and Shannon for the admixture in vitro work. And lastly, I just want to make a plug for we are hiring. We're, as Simon mentioned, I'm really excited. We've started a new initiative at University of Minnesota, where we're really working to leverage mechanistic mathematical modeling and engineering principles to optimize therapy development. We're doing, we've got initial partnerships in a lot of cell therapies at the moment. So particularly if you have an interest in cell therapies and immunotherapies, please consider emailing me or the center for more information. And I should say the center is in collaboration with Kevin Leader and David Odie from systems engineering and biomedical engineering. So really an interdisciplinary team. Thanks.
Excuse me, I don't have COVID, I just have a cold. I tested this morning. One of the penalties of flying around the planet. Anyway, any questions?
What is the value of F between the two phases of the power law, do you think?
The value of
Well, you say that at some point the power law transitions from 1/2² to 1/2.
oh yeah, like where does it actually? Yeah, that's something that we started doing some investigation into, but I don't have off the top of my head what we came up with. So that's a good question though. We did look at that. It didn't make it into the paper, and I've now jettisoned that from my mind. I apologize.
So thanks for the talk. I wonder if you played around with the model, if you can lend experimental support to the theoretic Gaussian approximation, Gaussian process function.
You mean for which part of the talk are you?
So this is based, if I understood you correctly, this is based on the Gaussian process.
The SFS?
The derivation, or is it separate?
So these results, the SFS results, are a direct analysis of the branching process model. This model. So there's no Gaussian process approximation in this part of the talk.
Okay, no, fine, thank you.
But the same question applies to the first bit though, which did have the Gaussian process
There,
thing in it, which I think presumably needs some looking at.
okay, so you're just
Okay,
discussing.
Okay, fine, yeah.
Yeah. Yeah, so we did apply this to, so the question is, do we apply this to experimental data?
Yes.
Yes. So we did actually apply it to the same sets of in vitro data that I showed earlier here. Yeah, and so the stochastic population dynamic model does, you can see this like does pretty well with the deterministic model. We recover the same accuracy for the estimates. The error bars are improved for the stochastic population dynamic model.
So I was wondering if this may be like a very tangential question, for the first part, like the drugs were on some processes that probably controls the dynamics of the clones, right, at the division, delta, whatever. So would these results would hold similar for immunotherapeutic models where you have like cells actually interacting with the cancers in the culture?
Yeah, that's a really good question. I think in theory, yes, and we have been developing, as I mentioned, sort of these models with interactions between cells, and there's no reason we couldn't add immunotherapy or adaptive T cells, for example, into this into the model. It's sort of as you can see, the statistical framework is very simple, and you can change the population dynamic model as you wish to adapt to your situation.
I mean, I guess
think
like the stoichiometry of drugs, I'm not 100% sure how you're modeling the stoichiometry in the culture. Are you like assuming it's all saturating concentration, or is there a pharmacodynamic for each drug? If that is the case, that would be very different for the immunotherapy because cells can also grow and like shrink or whatever.
Yeah,
And that's like,
yeah. So I want to make it clear that we are modeling here what is happening inside that drug screen experimental platform. So we're not modeling, this is not an in vivo model, we're modeling the drug screen itself. And so that's a really valid point about immunotherapies, and that will call into question, or I think that speaks directly to the translational relevance of this kind of drug screen for immunotherapies. If that makes sense?
yeah.
So maybe it doesn't make sense to do it to do these sort of drug screens in that setting.
For your first talk, if you have two populations which is very similar parameters, when can you determine that there are two?
Yeah, that's a really good question. And we did a lot of computational investigations. There's, how close can the subpopulations get before you call them the same? Also, how small can the subpopulations get before you stop detecting them? So we did all sort of like a big battery of computational tests, sort of for the supplement of the paper. I guess I can't give you like a numerical answer because it's very dependent on all the other parameters in the problem. So yeah, I don't have much to say in terms of like, the IC50 or the GR50 has to be this close together before or this far apart before you can tell them apart. But one thing that I will say is that the idea of doing a phenotypic deconvolution in terms of drug response is to really understand sort of like functionally speaking, if I give this therapy, like what's going to pop out as I kill the sensitive population, for example, like very sort of practical reasons. For those practical reasons, how much do we care if there are two subpopulations whose drug sensitivity is very close to each other? Then is it okay to think of them as one population? So that's sort of the flip side of that question.
Were you able to use the phenotype or the phenotype model at all to understand, I guess, any sort of relationship between like initial number of subclones in the population, so like initial level of heterogeneity and time to treatment resistance?
Yeah, so that kind of like this idea of using the heterogeneity profile as an initial condition in a model, a predictive model of, say, recurrence dynamics under a particular treatment regimen is basically what we hope will be one of the big uses of this. So, you know, when I kind of did this, we could imagine using this kind of predictive modeling to say, when will the M protein exceed a certain level?
Okay, but you haven't actually done any of those studies yet or developed those models?
We have done this a little bit in the multiple myeloma setting. But
Did you
not
see
beyond
like
that.
any general relationships between number of subclones and how long it takes to develop resistance when you do this in multiple myeloma?
Again, like it's gonna depend on what those subclones are. So if you've got a lot of resistant subclones and they're large, then recurrence happens faster. And but you know, having many sensitive subclones may not lead to faster resistance.
Is there anything online?
No.
Okay. All right, I think we're done. Thank you very much indeed, Jasmine.
Thank you for the question.